Voice check.
Mic check.
Hello, everyone. Welcome to Antibuddies. Today is our 28th Journal Club, and we have a guest today with us, Dr. Berenice Benayoun. Um, Dara, can you tell us a little bit about our guest today?
Absolutely. Dr. Benayoun is an assistant professor of gerontology at the University of Southern California, Leonard Davis School of Gerontology since the summer of 2017. And her lab's research focuses on the omic remodeling with aging and how these changes interact with biological sex to shape the aging process, specifically in the innate immune system. They are also one of the pioneering labs in developing the naturally short-lived African turquoise killifish, the Northobranchus fusari, as a new naturally short-lived vertebrate model for aging research. When she's not working, she enjoys playing strategy board games and home brewing meats and ciders. Can you tell us some of your favorite strategy board games?
Agricola, which is a farming simulation, which is actually a lot of fun. I know, like, some people's downtime, idea of like ideal downtime doesn't involve crunching numbers, but it can still be fun.
Hold on. What's the name of it? I'll write down.
Agricola, Agricola.
Okay. This is one of my favorite strategy board games called Seven Wonders. Have you heard of it?
Oh yeah. It's a great game also because it scales well. Like 7 players doesn't take longer than 3, which is really nice.
Yes, that's the best part. Okay, so Berenice is going to tell us a little bit about neutrophils and how their biology is affected by age and sex. So, Berenice, without taking any more time, I'll hand over the virtual mic to you.
Thank you, Jayton, and thank you, Dara, for the introduction. And so, yeah, I'm going to tell you about a story that was published, I guess, last summer in July, where the lab was trying to figure out, using multi-omic strategy, what's going on with mouse neutrophils as a function of age and sex. And so this comes from one of the big questions we have in my lab, which is to really try to understand sex dimorphism in aging. And really, what I believe and what people in my lab believe is that with the traditional approach of most research labs, and not just in aging, not just in immunology, but pretty much that's plaguing all of biomedical science, we may be missing important parts of the picture in terms of what we know of biological processes. And so, again, I'm going to focus on what I know best, which is the aging field, but just looking at data sets where people have investigated what's going on with aging or in response to pro-longevity intervention. I can tell you that as of a few months ago, we haven't looked, last week, but as of a few months ago, about 80% of all published molecular studies, looking at transcriptomes or, methylome or whatever, tend to just look at male individuals. And when you talk to people about why they use males, there's usually a long list of good, quote unquote, reasons, like, oh, female data is so noisy, which is BS, by the way, or, oh, females are just males with hormones, which is also not true, or, oh, it's just too expensive to do both, all of which are not very good reasons. But the problem is that it results in us basically knowing very little about the differences in what's going on with aging or in response to longevity interventions between males and females. And so this is where a lot of people might be wondering, okay, but why should we care? Well, aging in particular is extremely sex dimorphic. I'm sure all of you are aware that, like, when you look at the life expectancy, women tend to outlive men by about 10 years. And that's true, in developed countries and not so developed countries. Women tend to always outlive men, at least in the human species, very clearly. If you go to lab models, the NIA, the National Institute of Aging here in the US, have been funding something called the Intervention Testing Program for the past 15 years to try to rigorously test potential pro-longevity interventions and see if they're going to work or not in mice. And of course, they found a couple of things that work, but the biggest things they found is that there's actually not a single pro-longevity intervention that works the same in both sexes. Basically, everything they found either works much better in one sex than the other, or it works only in one sex to increase longevity and health, and not in the other sex. And what that tells me is that, if aging was the exact same process in both sexes, then in theory, the exact same intervention should have the exact same impact. And the fact that it doesn't means that there's going to be some fundamental differences in the way females and males age, which are really important if we want to get to a point where we can leverage this to help improve human health ultimately. There's other, indicators that aging is going to be really different between sexes. In humans, menopause is a huge, actually, health event in the life of women. A lot of people tend to dismiss menopause as just like, oh, it's when women can no longer have children. But actually, it is known to be the single biggest predictor of how healthy a woman is going to be when she ages, with all the age-related diseases you can think of, like, osteoporosis, cardiovascular disease, Alzheimer's, all of those skyrocketing after menopause. When you just look at the molecular level, what could underlie those differences? Well, it turns out that female and male cells are very different in the way that they regulate gene expression and epigenomic organization. In fact, the NIH Roadmap Epigenomics project, which was looking at epigenomes across different tissues, different cell types, and they had males, females, they had different disease states, they had different ages. Well, the largest differences they were able to see, and they were very surprised about this, was between the male and female samples of tissues, even when you were looking at somatic tissues like the liver, for instance. And there's several reasons where this may be coming from. When you observe sex differences, the one that people immediately think of are, hormonally driven differences. Estrogens and androgens have somatic targets. And so since those are transduced to nuclear receptors, you can get, local chromatin reorganization when they're signaling with them. But what people don't necessarily think of as often is that just the sex chromosome karyotype can have impact, because even though one of the X chromosomes is inactivated in female XX cells, there is actually 10 to 15% of the genes on that inactivated chromosome that escape inactivation and are expressed in a biallelic fashion in female cells. And those include transcription factors and chromatin remodelers. And so that could explain a lot of why there are fundamental differences in the way gene expression is regulated. And finally, more relevant to this audience, the immune system itself and immunity is very sex dimorphic, even if we take aging out of the equation there. For every man that has an autoimmune disease, you can find 9 women with autoimmune disease. And sort of on the other end of the spectrum, men tend to have much worse outcomes in response to infection or sepsis. And, again, the current pandemic, COVID-19, has shown that 3/4 of the very severe cases are men. And so, that kind of immediately shows that, okay, well, the immune system is going to be very sex dimorphic. We know aging is very sex dimorphic. So this is sort of very interesting nexus of figuring out, okay, how are those two things going to be interacting? And so this is where, when I first started, and I'll give the shout out with a picture in a couple slides, but I had a friend from my postdoc day, Shalina Taylor, who's a neutrophil buff. And she was saying, okay, well, we need to look at neutrophils. People don't look at them enough with aging, and, see what happens. And I have to confess that when she told me that in 2017, I was like, neutrophils, well, okay, I know I've seen those on my blood test, but I don't know much about them. But since then, I've actually grown to respect neutrophils tremendously as basically super cool cells. So for those of you who might be in the same situation I was a few years back, what are neutrophils and why are they potentially interesting and challenging to work with? So this is an H&E staining of neutrophils, where you can see they have this really interesting nuclear structure, polylobular nuclear structure, which basically earned them their membership into the class of cells called polymorphonuclear cells. So this is actually one nucleus, but that has an extremely compacted chromatin structure that gives it this weird appearance. Now, neutrophils are extremely short-lived cells. On average, once they finish terminal differentiation, they're going to live something like 12 hours, even though in some cases, especially if they invade tissues, they can live a few days. But for sure, neutrophils are not as old as the organism that they're in. They're extremely short-lived cells, which makes working with them in general a pain, because it means you need to do any functional assays extremely soon after isolating them. Now, what's really cool about them is that they've evolved to be able to weaponize their chromatin. So this is an electron microscopy picture of neutrophils who have extruded their chromatin. So here you can see those fibers, which is a process called netosis, because they those chromatin fibers in this case are called neutrophil extracellular traps, or nets. And those, as you can see here, can trap pathogens because chromatin is really sticky. And so here you can see those little bacilli that are trapped in the nets. And the ability of neutrophils to undergo netosis is actually crucial in the first line of response to infection, because they can immediately immobilize pathogens and help kickstart things, and then macrophages can come and phagocytose those nets and process them. And really, it's actually quite clear that not having functional nets is incompatible, with life for mammals. So we need functional neutrophils. Now, the problem, though, is if you make nets for no good reason, this can actually promote disease and inflammatory disease in general. So there's sort of a really delicate balance where you need your neutrophils to be able to go berserk when they need to, but at the same time, if they're too active, you have potential health issues. So with all of that, basically Ryan, a very talented graduate student in the lab, helped notably by Minhu, a postdoc in the lab, did a lot of work to try to figure out what was going on here, with the help of, again, my former postdoc friend, Shalina Taylor, and Kevin Contrepoix at Stanford University, who helped us, with the, some info on the neutrophils and with the mass spec aspects of the multi-omic profiling study. And so, yeah, basically, I'm gonna now tell you about what we found in our study, doing this multi-omic profiling. So the idea there was that we could take advantage of the fact that the NIA has an aging mouse colony where everything is like very well controlled, where we can get mice from that same colony at the ages we want from both sexes. And so we got two different time points, a young time point at 4 months of age and an old time point at 20 months of age. And again, for those of you who are not necessarily in the aging field, we choose 4 months as a young time point because that's actually about where hormonal cycles for mice are stable. And so, a lot of times when people use young time points of like 2 or 3 months of age, that's the equivalent of looking at a 14 to 18-year-old human, which is a little bit too young. Whereas for the old, we look at 20 months, which would be the equivalent of a 60 to 65-year-old human, so not too old. And that's also before the C57 Black 6, which is the strain we're using, actually tends to get pituitary adenomas, which can sort of muddle the sex hormone landscape if we look at older than that. Jayton, you have a question.
Oh yeah, just because, like you said, neutrophils, not a lot of people study that. That includes me. So I've always wondered if the lobes of the neutrophils and the fact that they spit out chromatin, is there any relation, or they are completely mutual things, exclusive things, I mean?
So from what I understand, part of the hypercompaction of chromatin helps with the physics of like throwing it as a net.
Okay.
But from what I understand, the different lobes might be useful. So there's two ways to do netosis. There's basically the netosis that ends up being cell death immediately, or you have what they call vital netosis, where only sort of part of it is expelled and the neutrophil can survive a little bit longer after doing that, which in and of itself is crazy. But again, I'm speculating here, but it might be partially because then you can expel part of your chromatin and, you know, keep some with the lobes. But it's probably also helping the cells to be able to go through like tinier spaces when they migrate to tissues to respond to infections. To have like really compact nucleus means they can go through smaller spaces as well.
Okay, thanks a lot.
Of course. So we had those 4 groups of mice, and then although in humans, actually I forgot to say that neutrophils represent 50 to 70% of circulating white blood cells, right? So you might think, okay, well, let's get them from the blood of the mice. Well, the problem is we can't get as much from the blood of mice, so we took advantage of the fact that there's a lot of them in the bone marrow, which gives us sort of a really abundant source for neutrophils that we can work with, especially to do omic type profiling. And so in this case, we used magnetic purification, and there's a few reasons why we did that, and I'm happy to explain more if people have questions, but that allowed us to get very pure neutrophils from the bone marrow. We're between 95 to 98% purity in a very sort of fast and parallel manner. And then with those purified neutrophils, we did a 3-pronged multi-omic characterization, looking at the transcriptome by RNA-seq, the metabolome, and lipidome by mass spectrometry. And so what did we see? And so again, one of the big things when you're doing omic type analyses is that the human mind really cannot fathom something like, you know, if you have 16 samples in this case times 20,000 genes, a 16 column times 20,000 row matrix makes no sense to a human brain. And so one thing people who are doing omic studies like to do is to reduce the dimensionality of the data, to project it in a 2D space, which humans are really good at interpreting, to see if there are big things that separate your samples. And so you may be familiar because a lot of people use PCA, or principal component analysis. We prefer in my lab to use MDS, which is multidimensional scaling, because of the way it handles, basically the way it handles the residual variation. Again, I don't want to bore you with the technical detail, but the end result is the same. You can project your data in a 2D space to see the big axes of variation. And so what would we notice here? So the first thing we noticed, and so I'm going to use the same color code for the entire presentation. The pink represents the female mice, the blue represents the male mice, the lighter colors represent the younger mice, the darker colors represent the old group. The first thing we noticed, kind of to our surprise, because we were expecting aging to have a bigger impact, was that we could very easily separate in that 2D space the female and the male samples without problems. The aging axis you could sort of see, so here in the diagonal maybe, where the young samples are more clustered in the lower right corner and the older samples are more in the upper left corner. But it wasn't like as clear of a separation. And so consistent with this, we are able to see genes that are differentially expressed with age and with sex at the transcriptome levels. So if we look at the other layers now, the metabolomics, here, again, we're able to very easily separate the female and the male samples. But what was more surprising to us is that you can actually see that although in each group for sex, so here the female samples or the male samples, you can separate the young and the old. Globally, there's no general direction for aging, with both males and females. So for the females, it's sort of separated in the first dimension, whereas for the males, it's separated in the second dimension. And so consistently, we do find metabolites that are differentially abundant between females and males, but we do not find metabolites that are generally differentially abundant with age. And the reason we can't do it here sort of in a separated sex fashion is because metabolomics is very noisy. And with n equals 4 only, without being able to take advantage of, you know, the 8 samples per age and per sex, we don't have the statistical power to get things specifically in one sex and not the other for metabolomics. And so we see metabolites different with sex. And so we see a very similar picture when we look at the lipidomics, where we can again separate the female and the male samples pretty clearly, but the aging axis is not clear. And again, similarly, we see a lot of lipids that are differentially abundant as a function of sex, but basically nothing happening as a function of age. And what's interesting for those of you who might be neutrophil enthusiasts, for the lipidomics, it actually showed us that the male neutrophils seem to be holding a lot more lipid droplets than the female neutrophils. And that's something we're following up on. But since my lab is an aging lab, I'm going to talk more about the aging stuff. So I'm not going to come back to the metabolite and the lipid for the rest of just the purpose of this presentation. So if we look at the gene expression, so here we have clearly a difference in the basal state of the neutrophils between females and males. But when you're thinking about, sex differences in aging, there can be two things that happen even if you always have a different starting state. You can have a different starting state and the same trajectory during aging, so the same changes occurring in the males and the females, even they started from a different place, or you can have different trajectories during aging. And so to measure this, in this case, we look at the rate of change in gene expression for every gene that we can detect as expressed in the transcriptome with aging in the females, here in the x-axis, versus in the males, in the y-axis. And the idea is if the aging changes are very similar, then this should basically be almost all on that red diagonal with a very good correlation. And so when we look at the rank correlation row, the Spearman's row, the correlation is really good. It's actually so good, the computer can't even calculate a p-value and it says it's 0. So very correlated changes, which means the transcriptional aging trajectories are very similar between the sexes. Although it's not in the paper, I just want to highlight that we've done this kind of analyses in other cell types. And I can tell you this is not always the case. There are cell types where the trajectory is very different with aging. And so here it's very correlated, so it seems the trajectory is very similar. But then if we look at the genes that pass significance in one sex and not the other, we see that there's a lot more that passes significance in the males, in this case with age, but not in the females. But if you look at them, it's not as if they're completely uncorrelated from the females, it's just that they look like they have a bigger slope. And so the way we interpret that is that there's some sort of acceleration of transcriptional aging for the male neutrophils compared to the female neutrophils. And we're not quite sure what that means functionally, because for the functions we've looked at, you will notice that we actually see the females having more drastic changes. But we have all those changes. We have things happening with age. We have things happening as a function of sex. What are the biological functions, right? It's nice to have lists of genes, but what we are interested in as biologists is, okay, what does it mean functionally? What's going to happen to those cells and their function? And so to do this, we're using a way of measuring functional enrichment called GSEA, or gene set enrichment analysis, where you can basically rank all the genes in your transcriptome from most upregulated to most downregulated to see if there are processes that are enriched at like the top or the bottom of your list, so tending to be upregulated or downregulated. And so, like I said, we can take full advantage of the fact that the trajectories are very correlated between the females and the males, to look at aging while regressing out the effect of sex. And of course, and that will be the next slide, sex while regressing out the effect of aging. So what are the big pathways that are regulated with aging? So here I'm going to show you the top 10 most upregulated pathways with aging and the top 10 most downregulated pathways with aging. So when we look at the upregulated pathways, there's a couple of things that make sense to people in the aging field, like autophagy is something that is known to be misregulated. It's one of the hallmarks of aging, actually. We see FOXO-mediated transcription. FOXO is a longevity gene. So, I mean, that kind of makes sense, but there's nothing too like waha moment on the upregulated side. But then if you look at the downregulated side, this is where things start to be kind of weird. We noticed immediately that there's a huge enrichment for terms related to cell cycle and chromatin. And again, in case you didn't know it, neutrophils are completely post-mitotic. They're not proliferating. They're completely terminally differentiated cells. So that's kind of weird. And the reviewers actually for the paper were unconvinced that this was the case. So they made us check. And I can confirm to you, neutrophils are indeed post-mitotic. We had like 95% of cells in G1. And no difference in those proportions across the different groups. So this is not because there is differential proliferation. Now, what's interesting is that there's some data from other groups suggesting that the cell cycle machinery is co-opted in neutrophils to regulate mitosis. So, you know, that makes sense because of the whole chromatin metabolism aspect of cell cycle. But basically, we see that this one of the major age-related functional categories is that chromatin theme. And so now when we do a similar analysis to see what about the genes that are female biased versus male biased, so for those sex differences, again, showing top 10 most female biased categories and top 10 most male biased categories, you can see that on the female side, you know, the diameter of those circles is much smaller, showing you that it's actually, there's less things that are very enriched on the female side. Things don't necessarily make a lot of sense. We're actually curious about the collagen, and we've seen a lot of extracellular matrix related things being female biased. So we're trying to figure out what that might mean. But again, in that specific paper, we didn't follow up on it. The one thing that I want to highlight, because it will resonate in a few slides, is that there is an increase in the interferon gamma signaling, which is normally an antiviral response on the female side. But then again, similar to the aging thing, when we look at the male biased pathways, we're again met with a plethora of cell cycle and chromatin terms, which again, not an actual change in the proliferation of the neutrophils, but suggesting that there might be something funky going on with the chromatin of the male neutrophils. And so, you know, with the transcriptome telling us that there is something going on with aging and sex with chromatin, then we were curious whether, you know, the chromatin architecture of those neutrophils would be regulated, or if it was just some weird quirk of the transcriptome analysis. And so to figure that out, we decided to take advantage of a technique called ATAC-seq, which is the assay for transposon accessible chromatin, which was invented by the lab of Will Greenleaf at Stanford, which a lot of people may be familiar with it because you can use it to identify changes in the local accessibility of chromatin, so how open a specific promoter or enhancer is. But what people might be less aware of is you can actually use the data from this to look at chromatin architecture, and specifically nucleosome architecture of the chromatin. And so we used an independent cohort of mice from the NIA, similar as before. We got our bone marrow neutrophils, and we performed ATAC-seq. And to figure out the chromatin architecture of those neutrophils, we used a algorithm called NucleoATAC, again from the Greenleaf lab. which allows you to derive from your TAC-seq data a certain amount of parameters that give you an idea of how open or closed the chromatin is. One of the first things it does is look at the local nucleosome occupancy. And so bear with me here. If you've never thought about these things, when you're doing these sorts of omic assays on a cell population, so anytime you're not doing single cell something, you're actually looking at a population average, which means that, although in one cell, the nucleosome would be here or not, when you're looking at a population of, in this case, 50,000 cells per sample, some of the cells are going to have a nucleosome there, some of them will not. And so you can measure sort of an idea of the fraction of the cells that had a nucleosome at that fraction. And obviously, the more often the cells have a nucleosome at that fraction, in general, means they tend to have a more compact chromatin. The more nucleosomes you have coding your DNA, the less accessible your DNA is in general. And so that's what occupancy of nucleosomes measures. You can also measure fuzziness. Again, this is at a population level. In each cell, the nucleosome has a specific position on the DNA fiber. But when you look across population, it's either very well defined and it's always at the same base pair all the time, or it can be a little bit fuzzy. And again, the more fuzzy it is, it means that there is ability to traffic it or move it around, which means that that part of the chromatin is probably more open if it's very fuzzy. And then finally, the last thing you can get from, again, derived from a TAC-seq is the interdiad distance. So the distance between two successive nucleosomes on the chromatin fiber. And so I think this one's the easiest to understand. The more distance between successive nucleosomes, the more open the chromatin is. So I don't want to bore you with like the bajillion box plots we have in the paper. So I'm just going to show you like the metagene analysis, which is consistent with all of the box plots to explain what we're looking at. And so for the purpose of this, I'm showing you the median nucleosome occupancy at genes that are expressed in the neutrophils. And so let's start for the sex effect. So here you're looking at the average occupancy of nucleosomes, like around the transcriptional start site. And in pink, again, you have the female, in blue, you have the male, and here the young versus the old. And you can notice in both cases, the pink curve is under the blue curve, significantly so, showing that there is an increased nucleosome occupancy in the males, regardless, compared to the females, regardless of age, which suggests a tighter chromatin structure in the male neutrophils compared to the female neutrophils. Now, if we do a similar analysis with respect to age, again, here females and males, you can see that the lighter colored curve is always under the darker colored curve. And again, significantly so. And so that means that with age, we have an increased nucleosome occupancy regardless of sex. And so it looks like we have that tighter, chromatin in males and with age. And again, I don't know how many of you are familiar with, aging research, but that is immediately weird to people who are doing aging research, because one of the main theories of things that drive aging is notably something that's called the loss of heterochromatin with aging, with the idea that the chromatin becomes looser and that you have like more weird cryptic transcription and everything. And so it looks like neutrophils are actually going against the grain of what every other cell is doing with aging by, instead of having looser chromatin, they have tighter chromatin. But again, remember, in neutrophils, chromatin's not just about gene regulation. Chromatin's about function, because that's how they can do mitosis. And so this could mean that there's going to be some functional consequences. So now this is with the TAC-seq. And because this was sort of anti-intuitive, the aging part for us, we were like, okay, so what can we use as an independent measure of global chromatin opening or closing to see if this is just, maybe we mixed up our samples or something or not. And so we went back to the RNA-seq data, which remember was an independent cohort. to see whether we could see differences in the transcription derived from repetitive DNA. Because indeed, so, the human genome's about 50% repeats, the mouse genome is 37% repeats. Normally, those are extremely well repressed because when they're expressed, some things can happen that aren't good, notably because a lot of those repeats are transposon derived, and those transposons kind of look like viruses. So, the pattern recognition receptors in the cell can recognize them as if they were viruses and get an antiviral response going. So you might remember I alerted you to the fact that we had this interferon response signature in the female cells, right? Well, if we look for differentially expressed repeats in our RNA-seq, what we saw was that we could get very clearly aging regulated repeat elements. And so here, young, old, you can see that the majority of them seem to be going down with age, which is consistent with a tighter chromatin structure in the older neutrophils. And then when we look for sex regulation, you can see that very clearly the female neutrophils have higher transposon and repeat transcription compared to the male neutrophils. And so that's completely consistent with what the ATAC-seq told us, even though, at least for the aging part, it's sort of the opposite of what we were expecting. But there's definitely something going on. And here, the tighter chromatin structure that we found by ATAC-seq does correlate to lower repeat transcription. And so again, I mentioned before, chromatin is not neutral for neutrophils. It's part of how they do their immune response by netosis. And so we decided to measure the netosis capacity of neutrophils from the different groups using a simple flow cytometry-based assay, taking advantage of a cell impermeable DNA dye, Cytox green. And the idea is if the cells are healthy and have not netosed, Cytox green can't get in. And so the cells, healthy unnetosed cells, will not be green, whereas the cells that have undergone netosis, they will have extruded their DNA, and so they will come up as being green. And so in this case, we have a control condition, or we induce netosis using a chemical inducer that makes the neutrophils believe something's amiss. We wait 2 hours, and then we look for the induction of netosis in our samples by looking at the PMA versus DMSO green cells. And so this is kind of noisy, and there's a lot of reasons for it. But what we noticed was really interesting and showing again the importance of doing both sex and age together, because the interaction of those factors sometimes is not sort of what you would expect it to be. So in this case, let's start by looking at each sex separately. When we look at the females, you can see that we have a very significant increase in the netosis induction capacity of those neutrophils with age, so between the young female and the old female. When you look in the males, although the median is a little bit higher, there's really no significance. So it looks like there's not much happening in terms of the netosis capacity of the males, although that might be because of the very large standard error that we observe. In the young, and this is subsignificant because the data is noisy, but we do believe for other reasons that this is real. The young females have lower netosis capacity than the young males, although this is flipped in the old animals, where the old females have the highest netosis capacity of all the groups. And so that does mean that we have a very strong age by sex interaction. And that, if you only looked at the males in this case, you would have been, well, there's nothing happening here, and, let's move on. Jaitin, you have a question.
I wanted to comment that this is such a big deal right here, that you're showing that these females, they have more netosis with age, because as you mentioned earlier in the presentation, that there are a lot of autoimmune diseases that are biased towards females, and a lot of these rheumatic diseases like lupus, they have this anti-DNA component, and if you have these neutrophils spitting out their nets easily, that explains a bit.
Yes, exactly. Yeah, very good. Good point to make. Thank you, Chetan. But yeah, so, again, the importance of looking, at everything and not being like, well, it's too expensive or it's too complicated. So we see all of those, differences. And so one of the things you can do using the multi-omic type approach we have is try to see if we can identify candidate drivers of what's going on in terms of age-related or sex-related differences. Here, for simplicity, I'm just going to talk about the aging side of this analysis, but we did a similar one on sex that's in the paper. And so here we decided to take advantage of machine learning to do this. And so machine learning is really when you give the computer a bunch of information for the computer to learn predictive features that are linked to a variable of interest. And I know that sounds very esoteric. Think of it as, you guys have Netflix. You tell Netflix, I like this movie, I like this TV show, and then Netflix says, okay, based on that, we think you're going to like this and that, right? So here we're not trying to learn, favorite movies or favorite TV shows. We're trying to learn whether a gene is, upregulated or downregulated with age. And so in this case, this is what we're trying to learn, not like, but up with age, down with age in neutrophils. And then for every single of those genes, we can collect information. So, we can collect information about the genomic environment of the gene, what's the CG percent, what's the CPG coverage, these kind of things, the length of the gene, the number of exons. So really just based on the sequence, right? Since we performed ATAC-seq, we can get information about the local chromatin accessibility at the promoters or enhancers of those genes. And then finally, taking advantage of the fact that, so many labs around the world have been trying to figure out targets for transcription factors and things like this, we can actually use those ChIP-seq, ChIP-chip, cut and run, all of those data that are there to have a bona fide list of whether like any specific gene is a target of any specific transcription factor. And so then we feed all of that information to the models, to the machine learning algorithm to learn models. One thing that I want to say is that if you give data to a computer and say learn, it's always going to spit out something. It's never going to tell you I couldn't learn anything. The problem is sometimes the model it learns won't be better than a coin toss. And so it's important to always check on data you didn't use for the training of the model, whether your model is better than a coin toss. And so in this case, we do have models that are much better than coin toss, over 65% accuracy, suggesting we are capturing biological signals. And so only once you know your model is not a coin toss should you go and try to interpret your model. And the thing that's nice about the types of models we used is that, I didn't put a black box here because they're not black boxes. We can actually go back inside and see what helped the models reach an accurate description of the data. And so these are two types of models we use, random forests and gradient boosted machines. Again, you don't need to understand how they work, just that they use the information to make the prediction. And so here, the darker the red, the most important the feature was for the model to make an accurate prediction, and the bigger the diameter, the most important the variable was. And so in the end, it ends up being a combination of those. And so here we ranked the top 20 most important variables combined for those two types of models. And so we see a lot of interesting things, including, that here the CG and CPG percents are very important. And we think this may have to do with DNA methylation, although we didn't measure it, because it's actually very well known that you can predict the age of samples based on their methylation levels. And so the fact that it looks like the percent of, methylatable CPGs around the gene might be important to predict age-related changes in gene expression is not completely absurd. Then among the very important ones, how open the chromatin was, or how much the chromatin accessibility changed with age, were also very useful to predict whether the transcription of the gene would change with age or not. Again, this is sort of expected. Now, what's interesting is if we look at those transcription factor target status things, some of them we don't necessarily make sense of, but some of them might be really interesting. I mentioned earlier, FOXO family transcription factors are known mediator and regulators of aging. And so the fact that being a target of FOXO is a good predictor as to whether you're going to change with age or not in neutrophils, that's pretty interesting. Or here, SIRT6, which is a NAD-dependent histone deacetylase. What's interesting is SIRT6 is actually one of the very rare things genetically that we know if it's knocked out, the mice undergo a progeric phenotype, so they have accelerated aging. If you overexpress it, the mice are long-lived. So it's a very clear regulator of aging. And what's interesting, and again, I'm not showing it, but I can tell you, we also find it as a big predictor of sex differential gene expression, and SIRT6 is actually sex dimorphic in the longevity effect, where it affects males. Depending on the papers, it only increases the lifespan of males, or it increases the lifespan of males much more dramatically than that of females. And so we're actually trying to follow up on SIRT6 as a potential regulator of those phenotypes. But the idea is here we are presenting potential mediators that can be mined for people to figure out what is driving those differential phenotypes.
For STAT5A, do we have any hypothesis? Is it known to have an effect on aging?
I mean, aging, one of the big things, which is a word that some people hate, but I'm still going to say it because I think it's nice. There's this concept of inflammaging, where, there's a lot of data suggesting that abnormal activation of inflammatory pathways is actually driving a lot of aging phenotypes. And so STAT5A, in that signaling pathway would make a lot of sense.
Okay.
But for aging itself, outside of the inflammation, that doesn't mean there's not something. It just means nobody's looked at it yet.
Okay.
Yeah. So, we have all these omic. One of the things that's also nice is if you can predict new biology that people had no idea about using that omic. And so for this specific thing, we went back sort of like, and this is for those of you who might be dabbling in omic analysis, this is a lesson of sometimes don't just look at the ensemble data, but go back and just look at the list of genes. So in this specific case, this whole, the last part of the paper came from just us looking at the list of genes, from most male biased to most female biased, and noticing that there was something really weird in terms of the top 25 most male biased genes in the neutrophils. So again, we mentioned mitosis, but another thing that neutrophils have in their arsenal, an antimicrobial arsenal, is that they're packed with granules that are full of proteolytic enzymes and antimicrobial peptides. And there's different types of them. There's at least 4 types of granules. They're zoophilic or primary, and then secondary, tertiary, and accessory granules that have those very defined contents. And so when they don't induce mitosis, what they can do to still respond to pathogenic challenge is to just degranulate and release those proteases and everything in circulation to help defend against infection. Now, like everything in the neutrophil arsenal, it's a double-edged sword, because releasing too many of those proteases can have bad effects. So for instance, there's some data suggesting that ARDS, the acute respiratory distress syndrome, which can be sort of the final stage of sepsis, or actually that is seen in some COVID patients, is because some of those proteases can digest tight junction proteins, notably in the lungs, disrupting the seal of the alveoli and then getting water in the lungs, right? And people then drown in their own lungs. And so that can happen if you have too many of those proteases released. But what we noticed, again, when we were looking at most male upregulated versus most female upregulated, 5 of the known azurophilic or primary granule components were there at the top. And so this is a heat map showing known primary granule components at the RNA level, where we can see here, MPO, so meal peroxidase, the neutrophil elastase, elaine, proteinase 3, cathepsin G, which are like, again, hallmarks of those granules being very clearly enriched in the males compared to the females. But of course, that's qualitative, but we can measure whether this bias is significant or not, and it is. There's significant male biased expression for primary granule components. Of course, because neutrophils are extremely RNA poor, that could be some, weird transcriptomics bias. And so again, because the reviewers asked for it, we did some Western blotting, and the male bias is definitely there at the protein level as well in the neutrophils. So the next question then was, okay, what does that mean, right? If they have more of those proteases, including neutrophil elastase inside the neutrophil, does that mean we're going to have potential adverse effect in conditions where those neutrophils would degranulate? And so in this case, because aged animals are very expensive, we did this for now only in young animals. So we got 4-month-old male and female animals, and we put them in, and I'm not going to argue with immunologists. I know this is not the best model of sepsis, but it's one I can live with as the PI, where we just inject them with lipopolysaccharide for a few hours. So it's a sterile, septic signal to see what's going on. And we did this with sort of a time course, 1 hour, 3 hour, and 6 hour of exposure, LPS or PBS intraperitoneal injection. And then we sack the mice to get their serum and see how much of neutrophil elastase, one of those main components of those primary granules, had been released. So I know there's a lot here, so let me walk you through it. The first thing that you want to look at is the 0 hour of LPS exposure, which basically combines the PBS injected mice, and there's not really a time difference for the different PBSs, which is why we're putting them together. You can see that even without any LPS signal or anything at the basal level, the males have significantly more neutrophil elastase in their serum than the females, suggesting that there's already sort of a baseline of more proteolytic release in the males than in the females. Then if we look at the effects of the effect of injecting LPS over time, in both cases, both in the females and the males, and that sort of proves that we did inject what we think we did inject, we see an increase in the release of serum neutrophil elastase in the serum as a function of time in both cases, which again is good. It means the mice are responding to the challenge. Now, what's really interesting is at 1 hour, we don't see a big difference in the release, but as time progresses at 3 and 6 hours, the males have systematically more release of neutrophil elastase than the females. And we stopped at 6 hours because that's what we were allowed to do as per our animal protocol, but it looked like it was still on an upward trajectory for the males, whereas the females seemed to be stabilizing. And so, although I can't say that this is exactly what happens in humans, what I can say is that this seems to be consistent with what we know of men, in the clinic having worse outcomes for sepsis and a higher risk of getting ARDS when they have sepsis, right? We have some data that we did by reanalyzing publicly available proteomics data set on human neutrophils that seem to suggest that the extra loading of neutrophil elastase, et cetera, in male neutrophils versus female neutrophils is conserved in human neutrophils. So we have that increased elaine levels and release in the males. And so this is all I wanted to highlight for the journal club today. Hopefully you guys are convinced that neutrophil biology is very sex dimorphic, that the chromatin architecture of neutrophils is regulated with sex and with age, with potential impact on their ability to undergo mitosis. You can go to the paper to get all of the things I didn't mention because there's a lot I wouldn't have had time to. And what I can tell you now is that we're trying to figure out whether those differences that I've talked about are driven by the hormones or the chromosomes between females and males. And we have good reason to believe that this is actually almost 100% driven by sex hormones, which means that it could technically be a therapeutic handle in the clinic. And so really, in this era where everyone is talking personalized medicine this and personalized medicine that, and really thinking, oh, if you have this single nucleotide polymorphism, how should we treat you in the clinic? I would like for everyone to realize that we can use the biggest genetic difference of them all, a whole chromosome, as a first start to enact personalized medicine. And that maybe if we took into account, some patients are females, some patients are males, we could actually tailor therapeutics to make sure to take biological differences into account when designing these kinds of interventions. So I want to thank the lab, without whom none of the work could have happened. Sources of funding, because without funding we can't do experiments, and the collaborators for all their help. And I'm happy to take any questions.
Thanks a lot, Berenice. This was a wonderful talk. I have something to share. Until 2021, the number of aging-related papers I read in my life was 0. Okay. And then I came across this very nice paper from Matt and Raphael. I think you know which paper I'm talking about immunosenescence. And so that was my first experience with aging and how it relates to immunity. Because for me, I always assumed aging is a completely exclusive thing. It has nothing to do with what I do, which is talk about immune system. And since then, I have got a lot more interested in finding out how different variables are being affected. So first of all, I'm very glad that we do these podcasts and journal clubs about topics that we don't necessarily work on, but they help us in some way. So this paper was a big eye-opener for me, that you have these big differences. I wanted to ask you if you think other cell types might have these differences apart from neutrophils.
Yeah, I mean, I can, obviously we're not at the publication stage yet, but we are working on macrophages as well. We actually started working on macrophages. The neutrophils were the side project, but I'm sure you guys know sometimes side projects become main projects. But we have a lot of work of data on macrophages, which I want to say are even more sex dimorphic than the neutrophils. And this was what I was alluding to, where when I'm saying not only they start from a different state, in the young, but they also completely diverge with aging, the macrophages, where what's happening with aging in the male macrophages and what's happening with aging in the female macrophages is almost diametrically opposite, which is really interesting, but makes it really complicated to study. So that's why we're a little bit later on the macrophage story. But yeah, I can tell you that there is not a single cell type or tissue that we have looked at where sex differences aren't the biggest separator, among our samples. So I used to be surprised when people in the lab would come and tell me, there's those huge differences between females and males. Now I want to say I would be surprised if they told me there weren't.
Looks like I need to find out what's the sex of the cell line that I'm using. It's a macrophage cell line. I've never wondered if it's from a male mouse. It's raw. Yeah.
Those are males.
Okay, so that's another variable I need to worry about.
So the IC21 cell line is female, if you want to have both in your hands.
Oh, do you think these changes between sex, is it because of the chromosomal difference, or is it the hormonal part, or is it both?
Yeah, I wish I could give you just it's one of them, but it's both, and actually it's different depending on the cell type. So I was saying for the neutrophils, we know for sure we have preliminary data in a mouse model where we can revert the identity of the gonad in an adult, where we can reprogram an ovary to be a testis in an adult mouse. And so then these mice have testosterone levels identical to male littermates just for limited amount of time. When we do that, those neutrophils now look completely like male neutrophils, which is why we think the neutrophil sex dimorphism is fully hormonal.
Oh.
But we've been looking in brain cells, and in brain cells, in those same model where we can swap the hormones, the cells acquire a intermediate phenotype where they're sort of halfway between the female and the male cells, suggesting that there's an interaction. And so there's several models that allow you to look at those things. But yeah, basically I can't give you the simple it's all hormonal or it's all chromosomal.
Okay. Before I ask any more questions, Magda, do we have something from the audience?
We have so many questions from the audience.
Okay.
I'm very grateful for them, because they asked so many questions. And so the first one is, you told us that you can explain more about why did you choose Max for the selection of neutrophils. And we have a question, did you use positive or negative magnetic selection for your neutrophils?
Yeah, so we used negative selection, and that's actually one of the reasons. There's accumulating data, and I know with immunologists in the audience, a lot of people are going to be like, no. There's a lot of data showing that going through a flow cytometer actually completely changes the transcriptional state of cells because of the sheer stress in the flow cytometer. And so in general, in my lab, we try to avoid the flow cytometer because we know it's going to induce changes in the cells we're trying to look at. The other thing is when you're sorting cells from 20 to 30 mice, which is what we do for every experiment, for the flow cytometer, you need to do them one after the other. And from 30 mice, we would be there for 8 hours purifying our neutrophils. With Max, as long as you have the, the magnets, you can do 10 at a time, which is what we do, which allows us basically, if we start our dissection at 7 a.m., we have neutrophils by 2 p.m. from the 20 to 30 animals. So that was the main consideration. I think if you're working with a smaller amount of animals, then, the flow might be more doable. But unfortunately, genomics in general is super sensitive to batch effects. Which means that flow actually creates more problems than it solves. Hopefully that answers it.
Yeah, and there was a question about positive and negative selection, this magnetic selection.
We use negative selection.
Okay. Yeah. Okay. Yeah.
Because otherwise with the positive selection, especially with Max, is extremely stressful on the cells. So we try to avoid.
Yeah. Sure. And there was another question, and I'm thinking whether is it even possible to distinguish which genes are on which lobe of neutrophil nucleus? Is it even possible?
So it would be possible with using sorry, I can't remember what the, right now I'm blanking, but fish, basically, in situ hybridization. So you could figure it out. We obviously haven't done it, but you technically could, yes.
There are more.
Go ahead.
So there's another question about can you name any specific genes that you can remember that genes are and even metabolites which are characteristic for each sex, males and females? Could you name, or I should?
So for the metabolites, since there wasn't much, I didn't spend as much time looking at the gene lists. At the differential list. For the genes, I can name a couple. So there's so obviously neutrophil elastase is proteinase 3, MPO are higher in the males than the females. We see a bunch of interferon related genes. I'm sorry, my cat has decided to crash this. And then we have a bunch, and that's sort of again with the lipidomics, where we use triacylglycerols being enriched in the males and the diacylglycerols being enriched in the females. We saw ATGL being expressed at higher levels, if I remember well, in the females, which is what we think is leading to the fat utilization compared to the males. So yeah, ATGL is higher in the females. And those are the ones I can remember off the top of my head, but not more. I'm sorry, but the gene lists are available with the paper.
Yeah, so I referred them to the paper, so they're going to read about that. And there are 2 more questions. And do you think that the difference in transcription repeats could be linked to the chromosomes?
I mean, yeah, definitely. Basically, there are. It's just that the chromatin is generally looser, so the repeats are expressed at higher levels, which seems to be, based on the data we have, just genome-wide. It's not specific chromosomes that are de-repressing the repeats, and the fact that we see the interferon response in the female cells suggests that sort of a generalized thing going on with all those transposons being expressed and being detected by, MDA5 or other pattern recognition receptors in the cytosol, which creates that response switch. This is actually something that a lot of people are thinking about, in terms of what transposon transcription might be doing. And there's this hypothesis that I actually think makes a lot of sense, that transposon transcription could be sort of a priming event to put the cells in a state of alert, as if there was an infection. I'm sure you guys are familiar with the concept of trained immunity. So it's the same idea of sort of priming the cells. And because we know that the female immune system in general tends to be more active than the male immune system, then this increased transposon transcription in the females could be, that priming event that explains it.
And the last one, so are the macrophages that you are looking at circulating or tissue resident?
So we're looking at tissue resident peritoneal macrophages because they're the easiest resident macrophages to get. And I want to say we were recently vindicated. I don't know if you've journal clipped that one, but there's a recent paper showing that when you do dissociation of tissues, macrophage pieces tend to stick to other cells. And then when you purify on something like F480, you're actually getting the other cells because the macrophage membrane is with them, which creates a lot of issues. So we're really glad we've been looking at peritoneal macrophages because that doesn't happen when dissociation doesn't need to happen. But they are resident because we use naive, not thioglycolate elicited, which are the sort of monocyte derived ones.
So you don't get a lot without thioglycolate, right?
I mean, in a young, the young females are the ones we get the least out of. We get about 700,000 per animal. But for the old animals, we get like 1 to 2 million macrophages without elicitation.
Okay, just in case you're wondering why our cat is around, I'll tell you that there have been evidence that cats like our journal club because I had foster cats and they would always be watching the journal club. It's real.
They're just getting information so that they can take over the world.
Yeah, they want to know how their neutrophils are being affected. It's just, they're curious. They may not look like it.
I think that's the mice, like.
Oh yeah, there's the mice.
Because we're talking about mice, yeah, that's why.
All right, Magda, are there any more questions?
No, but I wanted to thank you very much for your answers, and I want to thank our audience because these questions were really great. Yeah,
thanks a lot. And I think we are close to the ending of our session, so unless you guys have any more questions, I think we can wrap up the session.
And I just want to say, if anyone has like questions later, that the paper doesn't answer, feel free to reach out and let me know. My email is in the paper. I'm the corresponding author. So
yes, please feel free to reach out to Berenice. And with that, I think we'll end our 28th Journal Club. Thanks a lot, Berenice, for being here. And thanks a lot to the audience. We'll be back in a few weeks with another Journal Club. Until then, see you all. Bye-bye.
Bye.